This recording is going to cover the process of gamete production in the female, also referred to as oogenesis. Now remember, this is a result of meiosis, again, which is a biological process by which diploid cells undergo cell division to form haploid gametes. Now, oogenesis is a little more complicated than spermatogenesis in the male. Women, we're just a little complicated. Now, the process of oogenesis is going to take place in the ovaries, which you see right here. And just a quick reminder, since I'm here, is when the egg is ovulated, it will travel down the fallopian tubes and implant, if it's fertilized, in the endometrium of the uterus. And typically women alternate which sides they ovulate from, but I could be wrong. Now on the ovary, the outer part of the ovary out here is the cortex, and that's where we're going to see we're going to be talking about these various structures as we go along. Now, the process of oogenesis actually begins before birth. It occurs during fetal development. You do have an oogonium, which is diploid, but they're going to begin meiosis 1 during fetal development. And you have a primary oocyte here, which is still diploid, and which we're going to say here, this is the primary oocyte. This would be the oogonium, which is diploid. So both of those are diploid. Now, the primary oocyte is going to begin the process of meiosis 1, but it's going to become suspended. It will not complete it. That will not be completed until after puberty. So it begins the first phase, prophase 1, but it doesn't complete. It doesn't go through the entire stage. Now, what happens at puberty, once a month, a primary oocyte, which we see here, will then, and we're going to go through the steps involved, will complete the first meiotic division. But what's unusual in the female, it's not really unusual, but it is to me a little unusual, but it's cool, is that you have an incomplete division of cytoplasm. So you get a secondary oocyte, which is haploid. So here's a secondary oocyte, which you see here. So that's one. And what we refer to as a polar body. Now that polar body is not viable. It doesn't have a lot of cytoplasm. It's just a way to get rid of excess chromosomes. We're going to end up getting ultimately one viable gamete out of the process of oogenesis, unlike what you see in the male. So after the first meiotic division, you have a secondary oocyte. Well, that secondary oocyte will begin the process of meiosis 2, and that is what's going to be ovulated. Only if it becomes fertilized, and that's again, only if it becomes fertilized will it complete the second meiotic division. So you see here's the secondary oocyte that is becoming fertilized. Then it will complete the second meiotic division, and again you have an unequal division of cytoplasm. You get a haploid polar body, and this here is referred to as an ovum, which is still haploid. Not until the DNA from the head from the sperm and the DNA from the ovum mix does it become what we call a zygote. So the second the polar body from before I didn't draw it up on my picture, but that may or may not occur, which divides. But what you do end up with is you get 1, 2, 3, 4 haploid structures from that one diploid, because you have two meiotic divisions. But in females, we only have one viable gamete, and it's that ovum. Now, the reason why we have such a large cell, got a lot of cytoplasm, is because this egg, once it's ovulated, is going to have to travel about, not quite a week, but maybe 6-7 days before it makes its way to the uterus. So it's going to need a little bit extra fuel for on its way down to implant. Well, it is this, since I called it an ovum, it is fertilized. So it needs to be bigger, okay? So it's a much bigger cell. Now I do want to show this. If you don't want to listen to me talk, you can just pause this and write this down. But I want you to see some differences between spermatogenesis and oogenesis. Men produce lots and lots and lots of gametes because they start at puberty and they just keep going. Now females, they're born with a fixed amount of gametes. So they're born with primary oocytes. They are not born with oogonium, and they're not going to make any more. Okay, so it's a fixed amount and only a certain amount mature in their lifetime. In the males, you get 4 gametes for the spermatogonium, which is the germ cell. Females, you only get 1 viable one, that ovum. Now the process in the males I had mentioned in a previous recording begins at puberty, but in the female, this begins during fetal development. Males, timing, continuous, all the time. Females, it's once a month. We're going to go through a cycle, so we've got to go through that cycle, that ovarian cycle. In the females, when we hit menopause, when our ovaries fail, we're no longer can produce any more or functional gametes. Males is pretty much lifelong. And the timing of the gamete release. Men, you can release it just any day. Females, it's the day of ovulation, which is typically day 14 of your monthly cycle. Now, in the females, I had kept saying, is they have arrested meiotic divisions. So that first meiotic division, which was starting in fetal development, became arrested and it didn't complete till after puberty. And then the second meiotic division was arrested and it didn't complete unless the egg became fertilized. In the men, it's just uninterrupted. So there's some differences between the males and the females. But I do want to talk about, because it's an important part of oogenesis, is the ovarian cycle. What's going on in the ovary while this egg is developing? So we're going to refer to the events of the ovarian cycle. So typically the female has a 28-day cycle. Now I know there's some women with shorter cycles, some women with longer cycles, but we're going to assume typically a 28-day cycle. The differences between the lengths of some female cycles is usually during this part here. This is what varies. But once an egg is ovulated, if it's not fertilized, you will have your period within 14 days, give or take a day or so. So it's usually this part that varies. Now the ovarian cycle is broken up into 2 phases. From day 0 to 14, they refer to as the follicular phase, because this is where we're going to develop the follicle. Now these structures are referred to as follicles. Inside the structure is the various oocytes that are developing. From days 14 through 28, they refer to it as the luteal phase because of this structure called the corpus luteum, and we'll mention that. Day 14 is the day of ovulation, okay, where that secondary oocyte that was suspended in meiosis II is ovulated, but I want you to notice it's ovulated with some cells around it, and we're going to talk about that. So let's look at some of the things. So in the ovary, we're going to start, this is what you're going to see at birth, is this structure right here, okay? That structure is referred to as a primordial follicle. I'm going to have a hard time writing this. So a primordial follicle. What it is, it's a primary oocyte that's suspended in meiosis I that's surrounded by one single layer of squamous cells. Those squamous cells are referred to as granulosa cells. They help to nourish the egg and keep that egg suspended in meiosis I. What happens once a girl hits puberty each month, you're going to have that release of those gonadotropins, and specifically FSH is going to start to stimulate one primordial follicle typically. Sometimes you're going to get more. To stimulate one follicle to start to mature. And what's going to happen approximately over a day, 14-day cycle, this is what's going to happen, is that you're going to end up, those granulosa cells will start to multiply. And actually, we're going to start calling them follicular cells. The cells multiply. Actually, no, we're going to call them granulosa cells. That's the best way to call them. So the cells multiply, and we get, but initially what we have is a primary follicle. What this is early on, you end up with just one layer of cuboidal cells. And again, in the center of it is that primary oocyte kind of continuing along in meiosis 1. It's not finished yet. Those granulosa cells continuously start to divide and replicate, and you have here a secondary follicle, and I'm just going to use this term for secondary. The secondary follicle, and what you'll see with the secondary follicle is you see, again, a lot of layers of cells, of those granulosa cells. You will have a kind of thin layer that around the oocyte called the zona pellucida. You have that. You should have that in your notes, so I'll write it out. Zona pellucida. And it's going to be hard for me to do it, because it's going to just obscure the whole thing. It's just one little layer right around that oocyte. And that kind of separates the oocyte, which is still a primary oocyte, from those granulosa cells. And what you'll start seeing is at some point you're going to start to see what you see in here, and I'm going to fill it in, is you'll start seeing development of an antrum. It's going to be more obvious in this tertiary follicle. And the tertiary follicle, you see, again, here's the oocyte, here's the oocyte here. And you've got some layers of granulosa cells around it. You see some out here too. You have that antrum, it's fluid filled, and what's actually in that fluid is a lot of estrogens that these granulosa cells are producing. The estrogens are going to help with that development of that oocyte. The outer layer, which we didn't show it, but you'll have some more outer layers of cells referred to as the thecal cells, or the theca interna cells, which they produce androgens, but the granulosa cells then convert them to estrogens. The outer theca externa cells are just a fibrous layer of cells. When we get ready to ovulate, this thing's going to be very almost ready, looks like it's ready to pull apart. And here is a secondary oocyte. But the secondary oocyte is going to be surrounded by a few layers of cells, and those few layers of cells are going to be called the corona radiata. And I'm going to show you that in another picture, which is going to be a little bit better. These cells here are going to stay behind and develop into something called the corpus luteum. Now, the primary oocyte will finish meiosis 1 very close to when ovulation takes place. It will complete that, then you have your secondary oocyte. The secondary oocyte will then undergo the second meiotic division, and it's the secondary oocyte that's actually ovulated. Again, it is suspended in the in meiosis 2. Now here, these are actual pictures from the lab, so you can see what each of these structures look like. So these you're going to find way out there in the outer cortex. These are the primordial follicles. So you got one layer of squamous cells surrounding that primary oocyte. So the primary oocyte's right here. Here you see one layer of cuboidal cells. This is a primary follicle. Here's that primary oocyte again. You'll start seeing more than one layer here, and then this one, it's really nice. You see that pink right here? That's the zona pellucida. You can really see it well on that. This here is a secondary follicle. out here, what you're seeing out here, those are those thecal cells. Here is a tertiary follicle. It is pretty close to what we call a graphene follicle. I'm happy if you call it a tertiary follicle. A graphene follicle is getting ready to ovulate. So what you have here is this is what's going to be ovulated. This little structure you see here is called the cumulus oovarus. It's kind of like a stalk. And then here's that fluid-filled antrum. You see that, yeah, so this one is best for the fluid-filled antrum. All these cells out here is what's going to stay behind. But this is what's going to be ovulated. These cells out here, I'm just going to fill, actually, let's just put them in green. These cells out here is what's called the corona radiata. It's like a crown that radiates around that secondary oocyte. This is what these cells become in the second part of the ovarian cycle. This is the corpus luteum. So here, say this is the secondary oocyte that has been ovulated. In gold, I'm going to kind of highlight the zona pellucida. Okay, so there's the zona pellucida. The green is the secondary oocyte right here. There's the secondary oocyte suspended in meiosis II. And in blue out here, this is the corona radiata. Here's the sperm on their way to try to fertilize the egg. Now those sperm must penetrate through all these layers to get in here. So it's that acrosomal cap. It's going to start to weaken and degenerate. And when it hits here, it's going to start releasing those enzymes and substances, which can allow it to penetrate through these layers. Now there's some terms I want to introduce you to that they're kind of important. Is that acrosomal cap membrane has to destabilize so it can penetrate and get into that egg, but also you have to have these chemical changes in the tail to allow it to have greater motility, and that's the term called capacitation. So the sperm have the capacity to fertilize the egg. So capacitation is when you have the acrosomal cap membrane starting to destabilize, so we can be able to release those enzymes, get changes in the tail, so it's be able to start to move. Now remember, in the epididymis, in the male, it produced factors that prevented premature capacitation. Because what happens is shortly after those sperm become capacitated, they get that kind of whip-like motion, they hyperactivate, and they'll tire out easily, so we don't want them to capacitate too early. Now, another term I want to you aware of is something called the acrosomal reaction. This is when the sperm approach this yellow area, that zona pellucida, and it starts releasing its contents. So that's when it starts to dump those contents, so it's going to start to burrow in there. That's the acrosomal reaction. Now, once one sperm has snuck their little head in here, so I'm going to have a sperm, it sneaks its little head in here. What we want is we don't want any other ones. These guys, we don't want them in here because we don't want what is referred to as polyspermy. You want a haploid plus haploid diploid. You don't want the haploid egg plus haploid sperm 1, haploid sperm 2, and you get 3N. That's not viable. That's not a viable pregnancy. So what we're going to do is, once one lucky sperm has made its way in, is what's going to happen is there's something called the cortical reaction that will take place. That cortical reaction involves granules that are released from the egg, and I always say it kind of puts up a little brick wall, so no other sperm can get in. So that helps to prevent what referred to as polyspermy. So these are some terms that I want you to write down, okay? Now what will happen after the egg is ovulated, that those cells that remain behind will develop into something called the corpus luteum. So right here, this is your corpus luteum. Now what's allowing this to develop is actually luteinizing hormone. You're going to have high levels of luteinizing hormone and actually shortly before ovulation is the numbers of luteinizing hormone receptors have started to increase on the cells, these cells out here, so they're going to be very sensitive to luteinizing hormone, is LH levels are going to be fairly high during the luteal phase of the ovarian cycle. And you get development of this thing called the corpus luteum. Now these cells are referred to as lutein cells because they're yellowed, because that's what the term luteinize means. Those cells are going to produce high levels of progesterone and estrogen, but lots and lots and lots more of progesterone than estrogen. Progesterone is going to be important during this phase of the cycle, especially if there was fertilization that took place. Now, the thecal cells will continue to produce androstenedione and testosterone, but most of it, again, is going to be converted to the female sex hormones by these cells that stayed behind. Now, if the egg does not become fertilized, that corpus luteum will degenerate and it will become the corpus albicans. And I'll talk about what is needed for that corpus luteum to stay viable, because it's going to be important to stay viable if the egg becomes fertilized. But if the egg does not become fertilized, the corpus luteum will degenerate and become this corpus albicans. And it's about 12 days after ovulation unless fertilization has occurred. So we'll talk about what's going on in the uterine cycle next, which is going on the same time of what's going on in the ovarian cycle.